Good morning. Good morning, everyone. Good to see you. Happy Monday. Yes, please go ahead and type your name into chat for me. Thank you very much. Still waiting on a couple folks. I hope there's not too much background noise today. I have a fan running. It's very warm in here today. Thank you. Yes. If you haven't, go ahead and type your name into chat for me. And turn your camera on for me so I can see you. See all your smiling faces. All right. Well, I hope everybody had a nice weekend. We had some beautiful weather. Okay. So today we're going to talk about, I think, a fun topic, which is the fungi. We don't get to talk about fungus a lot in this microbiology class, but we do take some time today to talk about it in terms of some of the more practical aspects, the laboratory aspects of fungal microbes. So that'll be our topic for the day. I am going through your lecture exams. Those will be done later today. So look for that. Do take a minute or two to look at my comments that I've made on your exam, if I've made any. Most of you did very well. I was pleased to see. If you missed a question or two, I might have left you some comments to help you understand why you missed it. Maybe to give you some things that you can add into your notes, your lecture notes. So look for that. I'll be grading your laboratory homework tomorrow. I usually grade the laboratory questions on Tuesdays, so look for that tomorrow. And this week, of course, in your lecture, you have, remember, a difficult topic this week, the biochemistry topic, and also the topic of microbial growth and the mechanisms that particularly bacterial cells and archaeal cells use to divide. So that's coming up for you in lecture this week. There was a very interesting story in the news, in the local news, actually, I'll say regional news this weekend, which tied in directly with what we were talking about in lab last week. I don't know if any of you saw it, but down in Burlington, that's fine, Elaine, thanks for letting me know. Down in Burlington, Massachusetts, I know some of you are from originally from Massachusetts, there was a boil water order put in place. I believe it was Thursday or maybe Friday. And it was due to the discovery of our old friend, E. coli, during some routine water monitoring tests. So a very timely news story for us. Very interesting. So the poor folks down in Burlington had to boil any tap water that they wanted to consume, basically. So you could still shower, you could still, use it in the toilet, of course, things like that. But if you wanted to consume it, if you wanted to drink it, or you wanted to cook with it, you want to brush your teeth with it, any activity that would have you consuming it, they were advised to boil it. And again, that was because of a discovery of the bacterium called Escherichia coli, or what we like to shorthand as E. coli. So I've actually posted, I think, a nice news piece, a nice news summary of the story up on our discussion board called Questions, Questions, Questions. I'm going to pull that up for just a minute. So these are our discussion boards right here. And this is the particular one called Questions, Questions, Questions. And if you scroll down through the ones I've posted about unit conversions, resolving power, here it is. So this was down in Burlington, Massachusetts, and the State Department of Environmental Services, as well as the city of Burlington, put together this boil water order because E. coli was discovered in some routine monitoring. A couple of things that make this story interesting, I think, are number one, that this discovery was not made in the reservoir system that Burlington uses, that Burlington draws its water from, but rather on the clean side of their water treatment facility. So I'm not going to take the time to play the news story now, but it's right here. And what I'd like you to do sometime today or before our next lab, go ahead and come in here and click on that news story and take a look. I've also posted, if you're interested, this little fact sheet for you that they would have given to the citizens of Burlington as an instruction sheet, basically, for them to guide them about boiling water. And I have a question at the bottom of this little blurb I've written for you that I'd like you to see if you can answer. See if you can answer it without looking it up. Just give me your, best guess about this question that I've asked at the end. So this is up on our discussion board. Again, one of the things that makes this story kind of interesting is that this was not a discovery of too many E. coli in the reservoir itself. And Burlington draws water from two different sources, I believe. Don't hold me to that. But the water that was on the clean side of the system. So remember, we draw water out of these external locations, whether it's an open reservoir or an aquifer or some other type of water source. That water is brought in to a treatment plant, a treatment facility, where it goes through several steps of purification. And then it enters into the clean side, what we call the clean side or the potable side. And on the potable side, it's all closed. Nothing else is coming in. There's no open place. Nothing's exposed to the air. And all that clean water gets held in tanks, in storage tanks, so that it can be drawn out to the household as people turn their faucets on. So somehow this E. coli was introduced into the clean side, which is a relatively unusual event. And what happens is when that happens, you have to go through the whole system. You have to examine all the pipes, make sure there's not a leak somewhere. You have to check all your your joins between pipes. You have to check all the storage tanks. One of the most common ways that clean, treated water gets contaminated, as disgusting as this sounds, is if an animal gets into a storage tank and dies. So sometimes birds will get into these storage tanks if the hatch is left open on the tank. Rodents, small mammals, you name it. Things get into these storage tanks sometimes if the tanks aren't kept properly closed. So anyway, an interesting story based on what we've been talking about in lab, monitoring for the presence of microbes. So yeah, take a look at that today when you have a few minutes on our discussion board. Yes, Elaine, raccoons are kind of notorious for this. Raccoons are very curious animals. And if a storage tank, if the lid on the top of the tank, which is used to allow service people in to clean the tank and so on, if the lid is left open or ajar, a curious raccoon will go in there. And of course, once they get in, once they fall in, they can't get back out. So it's not uncommon for them to drown and then, have the carcass in there degrading, and all of the bacteria that's within that carcass then is going to be set into the water. And of course, raccoons, like all mammals, has E. coli in their intestine, just like you and I do. So yeah, this is why we test. This is why we monitor water. And we don't just test it before we clean it up and put it into storage tanks. We continue to test it in order to catch these problems as they arise. Oh yes, Marilyn. I actually just thought about that story as I've been talking. A terrible story about a human body that was put into a water storage tank, particularly a tank that was water storage for a building, an apartment building. And part of the reason that the police found this body is because the people in the building noticed that the quality of the water was degrading. And of course, this woman, I believe, well, I don't exactly remember the story, but I'm not sure they ever knew whether she was murdered or if she had committed suicide, but her body ended up in a water storage tank for a building. In a lot of cities, if you look around at all the high-rise apartment buildings, there are water tanks on the roofs. So a lot of particularly older buildings are serviced by rooftop water storage tanks. And unfortunately, those may or may not be monitored super closely. Yeah, so very, very important though, to make sure that our water is clean and free from potentially pathogenic microbes. All right, very good. All right, so I'm going to go ahead and pull up our slides and we're going to get started on our exercise about the fungi. And that's what you should be seeing on your screen right now, the title slide for the fungi. Now, our objectives for today are to talk a little bit about fungi and how we classify them. We're going to compare the fungi to bacteria specifically in terms of how they're the same and how they're different as microbes. We're going to talk a little bit about the saprophytic fungi, the common saprophytic fungi, and we'll talk about that term, saprophyte. And then we'll talk about some of the important features of the microscopic types of fungi or forms of fungi, which are the yeasts and the molds. Now, I have to say that the study of the fungi is very complex. You could take a course that would last all semester that would be just about the fungi. They are an amazingly diverse group of organisms, and they are very, very different from other eukaryotes. The study of fungi is called mycology. I have that word down here for you. Mycology. And again, you can spend a whole semester just talking about the fungal organisms. So we're only going to be touching on the fungi today, just learning a few things as they relate to microbiology. So fungi are eukaryotes. So remember, within the three domains, the bacteria evolved first, then the archaea, and the bacteria and the archaea are the prokaryotes. And the third domain that evolved was the eukarya. And the fungi are within that domain. They are their own group of organisms. They are significantly different from both plants and animals and protists. So they are their own unique group. Now, the fungi can be unicellular or multicellular. So that's why we learn about them in a class like microbiology, because they can be single-celled organisms, but they don't have to be single-celled. They also can be microscopic, but they don't have to be. There are both microscopic and macroscopic forms of fungi. So of course, by our lens of microbiology, we're interested in single-celled microscopic organisms. That's our definition of a microbe. There are single-celled microscopic fungi, and they include yeasts and certain molds. Now, there is a non-vegetative form of fungi, what we would refer to as spores. But these spores are different from bacterial spores, and I'll talk about that in a few minutes. Macroscopic forms of fungi, you've all seen mushrooms, for example, growing on the ground. Mushrooms are actually a reproductive structure for a macroscopic fungus. The most of the organism in a macroscopic fungus is actually under the ground in the soil. And we give that vegetative macroscopic organism a name. We call that mycelia. So a mycelium is the underground macroscopic form of a fungus. And of course, if it's macroscopic, it's also multicellular. From time to time, these underground organisms, these mycelia, will come to a point in their life cycle when it's time to reproduce. And that's when they will send mushrooms up above the ground. And if you've ever stepped on a mushroom out in the forest, you may have noticed some powdery material emerging from underneath the cap of the mushroom. Those are the spores, the fungal spores. Mushrooms are reproductive structures. So they are not the entirety of the organism. They're just a reproductive structure that the mycelia is forming at that moment in its life cycle. Yeasts and molds are microscopic, vegetative or cellular, microscopic forms of fungus. Now, one of the things that makes the fungi interesting, one of many things in my opinion, is that they get their nutrients through absorption. So they are literally going to absorb everything that they need from the environment right across their cell wall, right across their membrane. Remember that the cell wall inside of, or I should say, surrounding fungal cells is made out of a relatively unique kind of carbohydrate. Does anybody remember the name of that carbohydrate? Go ahead and type it into chat if you do. Remember, bacteria have peptidoglycan in their cell wall. Fungi have a different type of carbohydrate in their cell wall. Anybody remember it? Go ahead and type it right in. That's right, Connie. Sahara, good. Yes, this is chitin. Remember chitin? Chitin is the material that we find around the fungal cell in the cell wall, and also in the exoskeletons of insects, right? Which is why they crunch when you step on them. That's right. Good job, Valeria. Now, the fungi, they're going to take in whatever material that they need from the environment by absorption. So they're pulling the water, they're pulling the nutrients in across that cell wall. Chitin is a fairly strong carbohydrate. It's a relatively strong protective carbohydrate. It's not nearly as strong as peptidoglycan is, but it's strong. And like Valerie was saying, it's what's going to give chitin that crunch in an insect cell or an insect's body when it gets stepped on. So chitin is a protective material, but it is going to allow things to come across it, to come through it, so that the cell can absorb those materials across the membrane. Okay. Now, when it comes to metabolism, when it comes to the way that fungi get their energy produced inside of their cells, and we haven't talked about metabolism yet, but the majority of the fungi are aerobic organisms, just like you and I. So they're using oxygen for their primary metabolic pathways to make energy. There is a big exception to that, and that's the yeasts. So the yeasts are actually anaerobic. So they're using something other than oxygen in order to perform these critical metabolic pathways. Now, there are some yeasts that are aerobic, but there are a lot of other yeasts that are anaerobic. So you see both in yeast. For the rest of the fungi, the majority of them, the forms of fungi, the majority of them are aerobic. So the yeasts are a little bit different. The yeasts stand out from the other forms of fungi. And hopefully it makes sense to you that a great big macroscopic mycelium that lives in the soil, that's an organism that's going to use oxygen because it lives in an oxygen-rich environment. And when it sends its little mushroom structures up into the air, those cells are going to use oxygen because they're in an oxygen-rich environment. Some yeasts are also aerobic, but it's interesting that some of them, many of them, are anaerobic. They actually don't make use of all that oxygen. Yeah, Elaine's talking about stomping on mushrooms when you were a little kid, right? If you went out into the woods when you were a little kid and when you went on hikes, there are lots and lots of mushrooms that grow in New Hampshire, and some of them have that sort of puffer ball shape. You've seen them. And as kids, we used to love to step on them because you'd get a cloud of material coming out. And what you were releasing when you stepped on it were these spores. And I said a few minutes ago that These spores that the fungi make are different from the endospores that bacterial cells make, some bacterial cells. The spores that fungi make are actually their gametes, their reproductive cells or their sex cells. So something similar to egg and sperm in humans. So they're not survival structures for the fungus. They're the way that the fungus sends out its gamete, its egg or its sperm, in order to reproduce. Okay, so I said at the beginning of class today that I wanted to make some comparisons between the fungi and the bacteria. And we can really see some of these differences when it comes to food and food spoilage. The bacteria are one type of microbe that can be quite problematic for us in terms of food spoilage. It's something that we have to fight against when we process and package food. It's something we have to try to avoid in order to make foods have a longer shelf life. Bacteria can certainly spoil food, but the way that bacteria interfere with our food supply and the way that fungi do it are very different. And that's because these two organisms have very different sensitivities around environmental conditions. So, for example, compared to the bacteria, fungi can tolerate some conditions that the bacteria just can't tolerate. The fungi can tolerate much more acidic conditions on foods, and they can also tolerate less water. Now, I've got a picture here of a tomato growing out in somebody's garden. And if you've ever grown tomatoes in the garden, you've seen this on your tomatoes. This is actually a fungal organism growing on the tomato and spoiling this tomato. Remember, tomatoes are quite acidic. Bacteria just can't grow on a tomato like that and make use of the tomato as a food source. And the reason they can't is because of the pH of the tomato. Tomatoes are acidic. Now, they're not strongly acidic, they're only mildly acidic, but the pH can be down in the area of about 5 or 5.5 in a nice juicy tomato. And a bacteria can't survive on that. So whenever you see growth, natural microbial growth on a tomato or in a tomato product, that's fungal. The fungi, they don't mind the acid. They just don't mind it. The bacteria can't survive in it, but the fungi, they don't mind it. Now, the interesting thing about water, all cells on this planet require water. They require an ongoing, constant source of water, both the bacteria and the fungi alike, as well as all the other different organisms. But when it comes to food and food spoilage, the biggest difference that we see between the bacteria and the fungi is around something called water availability. So it's not just how much water is available to the cells, it's about how much of that water is available for them to use. Now, that may seem strange. Like, what do you mean? Isn't all the water available in a food? The answer is no. Sometimes in foods, the water is actually drawn up and bound to macromolecules. And the biggest culprit in this is carbohydrate. So If you take a food, a sweet food, for example, and one of my favorite examples here in New Hampshire is maple syrup. Maple syrup is a very, very concentrated source of sugar, right? It's delicious. We all love it. Very, very sweet, very thick syrup kind of material, right? There's water in maple syrup. There is, but there's also a lot of sugar, and that sugar will bind up that water and hold it in a way that makes it unavailable for the bacteria. Remember, polar things, you'll be reading and learning about this concept of polarity in lecture this week. Things that are polar are drawn to other things that are polar, chemically speaking. Water is a polar molecule, and it will bind with other polar molecules like carbohydrate. So carbohydrate is notorious for binding water. And even though there is water in maple syrup, most of that water is unavailable to the microbes. Bacteria have a higher requirement for available water than the fungi do. So you can see this. You can see this, and you may have experienced this. When you buy, a quart or a gallon of maple syrup and you bring it home, it is possible for that maple syrup to spoil over time, but it's not going to spoil from bacterial growth. If anything's going to happen, and it usually only happens if you leave it out on the counter, like a lot of us do, put it into some kind of a pouring piece of dishware and leave it on the counter, you might get some fungal growth on your syrup, but you won't get bacterial growth. And that's because the water availability in maple syrup is just too low. The bacteria can't survive in it. The other place you'll see this is in jams and jellies. I love jam. I always have plenty of jam around to put on toast and things. And jam lasts a good long time in the refrigerator, but it will spoil eventually. Jam and jelly are also materials that are wet. I mean, they have water in them, but they're also a concentrated source of sugar. So the water that's in that jar of jelly, it's mostly bound up to the sugar, to the carbohydrate. Bacteria, they have trouble growing in jelly and jam. They just do. So if you have an old jar of jam in your refrigerator, if it's going to spoil, it's almost always from a fungus. You'll open up the lid on the jar one morning and you'll look inside and there'll be some gross, green fungus growing on the top of it. It's almost always a fungus, not a bacterium. And that's because there simply isn't enough water availability in that type of very sweet, concentrated sweet type of a food. It's interesting. The fungi, they're okay. They don't have the same requirement for water availability that the bacteria do. So acidic foods, the bacteria can't really grow well in, and high sugar foods, high carbohydrate foods, the bacteria really can't grow that well in, but the fungi can. Now, the other thing to note is that if we switch this around, so instead of looking at the fungi compared to bacteria, if you look at the bacteria compared to the fungi, the bacteria can tolerate shifts in osmotic pressure better than the fungi can. Now, remember, we talked a little bit about osmosis in the beginning of the class. Osmosis is this process that drives water across membranes. And it's an effort by nature to get to equilibrium so that the inside of the cell and the outside of the cell have an equal concentration of dissolved materials, dissolved solutes. So the bacteria are better protected against these osmotic shifts of water than the fungi are. So fungi are better at growing in acid and better at growing in high carbohydrate foods, but the bacteria are better protected against rapid shifts of water across their membrane. And it's all because of the cell wall. Peptidoglycan is just more protective of that than chitin is. So the bacteria have an edge there. So Elaine's asking about botulism. One of my favorites. I know, I'm a nerd. So the organism that causes botulism is called Clostridium botulinum. And of course, as a clostridial organism, it's a spore former. It's also a toxin producer. And you're right, Elaine. You can, we used to have a lot more problems than we do now, but you can have a problem with botulism in canned foods. When you take foods and you put them into a can and you close that can up and remove the air from the can, the idea is that you're preserving the food in the can and it will stay fresh for a fairly long time until you open the can. This organism that causes botulism, there are two things that make it so dangerous. Number one, it's anaerobic. It doesn't use oxygen for its metabolic reactions. In fact, it finds oxygen toxic. So if any Clostridium botulinum gets into a can as it's being made, even after the air's removed and the can gets sealed, that organism is still going to survive. It doesn't need air. It doesn't need oxygen. So it can still survive. So it's critical when we can foods that we do it correctly. We get them to a high enough temperature to kill any botulism organisms that might be in that can. Now, like I said, with modern canning, it's a much less of a problem than it used to be in the old days. But you've probably heard the warning that if you ever pick up a can and it's bulging, the lid is domed, or the can is somehow bulging outwards, that's a can you want to throw away. Because the other thing that Clostridium botulinum does is secrete a gas. So we know that it's growing in that can, or we suspect it, because gas is accumulating in the can. Very, very dangerous organism. It creates a toxin, botulinum toxin, which will kill you. It's one of the most potent toxins known to man. So yes, you've got to can correctly, or you run into this danger of botulism. But here's the thing, more related to your question. We don't tend to can those super sweet foods. We don't tend to can jellies and jams. We tend to jar them, in glass. Remember, a can is different. A can is completely sealed. Yes, the jars that you put jelly in are vacuum sealed, but it's a different packaging system. So that's number one. And number two, botulism doesn't tend to show up in those foods. It turns out that most of the botulism that we used to see, again, from canned foods, most of the botulism came from canned meat and neutral foods. Now, what do I mean by that? Those are foods like corn and beans, like black beans and navy beans and kidney beans. Those foods have a neutral pH. Canned meat tends to have a neutral pH. So if any Clostridium got into those cans, that Clostridium is going to be able to grow because the acid level is not a problem and the sugar level is not a problem. And the Clostridium can thrive in that kind of can. Yeah, so canning is great. Canning has allowed us to preserve food long beyond the length of time we used to be able to keep it, but it has to be done carefully. And luckily for us, most of the canned foods that we consume now are produced commercially, right? They're produced by commercial, professional people, so they're much safer than they used to be. Oh, the other thing to know about botulism, just as an interesting aside, well, two things. Number one, botulinum toxin is the exact same thing as Botox. So yes, when people get Botox injected into their face, that's a very, very diluted form of botulism toxin. And number two, adults are much more sensitive to the organism. Babies are much more sensitive to the toxin. So in a food like honey, which is naturally produced, right, very high in sugar, it's not uncommon for the botulism organism to get into honey, but it's not going to survive, right? Because honey is too high in sugar. It's not enough water. The water availability is too low for the Clostridium to survive. But even a few cells for the short amount of time that they're going to survive, they're going to secrete toxin into the honey. So if you ever have a baby in your care, you would never want to feed a baby honey, a baby that's under the age of 1 year, because even though there won't be any Clostridium cells growing in that honey, there could be toxin in that honey. And even a small amount of that toxin will kill a baby. So yeah, it's interesting. You and I can eat the honey because that small amount of that toxin is not going to harm us. But a little baby can't tolerate that. So we tell parents not to feed their infants any honey. Yes, that's right, Connie. Under the age of 1 year, just don't do it. It's too risky. All right, very good. Good questions. Now, the real take-home message that I want you to walk away from today with is how diverse the fungi are. There are so many different types of fungi, so many thousands and thousands of genus and species in this particular group of organisms. And that means they have incredible diversity, genetically speaking. They have all different types of body forms or morphology, and they have an equally diverse number of different life cycles. There are some fungi, as I've said, that use aerobic metabolism. There are some that use anaerobic metabolism. There are fungi that take the mycelia form and produce mushrooms. There are other fungi that prefer to remain as a microscopic organism, like a yeast or a microscopic mold. There are some fungi that can be both yeast and mold at different times in their life. There are fungi that are sexually reproducing. There are fungi that are asexually reproducing. There are fungi that can switch back and forth between sexual and asexual. So I just want you to really hear the message that the diversity among the fungal organisms is utterly tremendous. They are so diverse, both in their structure and in the way their cells function. The only the real application to microbiology comes from a couple important forms of fungi that really impact us in terms of the natural world. So I want to talk just briefly about a couple of specific types of fungi that I'd like us to be familiar with. One of the when you compare fungi and bacteria, one of the things that hits you very quickly is that while these are two very different cell types, two very different organisms, I mean, one of them is a prokaryote, the bacteria, and one of them's a eukaryote. I mean, that's about as different as you can get. But when it comes to bacteria and fungi, it's sometimes hard to tell them apart on a microscopic level. In fact, it can be hard to tell fungi apart from all kinds of different microscopic organisms. A great example of this is a type of organism called Oomycota. This is the word right here. It looks like it ought to be pronounced mycota, but we pronounce that Oomycota. These are what we used to call the water molds. And the reason I even mention this is because this Oomycota organism is the kind of fungus that brought about a terrible moment in human history, which was the terrible famine that hit Ireland and, in fact, drove most of the immigration from Ireland to the United States back in the 1800s. There was a terrible outbreak of what was called potato blight across Ireland. And since the potato was a staple food for the Irish people, it created a famine that lasted several years, in fact. So, for a long time, we believed that the organism that was causing this blight on the potato, we believed it was a fungus. I'll show you a picture in a minute. It sure looks like a fungus. But we now know that it actually was not a fungus. This organism, this Oomycota, is actually genetically more similar to algae. But take a look at it and see how people got confused. Remember, the Oomycota are called water molds because you can find them in natural bodies of water. And here are some images of this type of organism growing in watery environments. And I think you can see why people thought this thing was a fungus. It sure looks like a fungus. It's very filamentous. It's very sort of fluffy, and it just has that fuzzy fungal look to it under the microscope. And when you look at it with your eyes, it also looks like a fungus. These are potatoes. This is the potato itself on the right, and this is the potato plant, the leaves of the plant over here on the left. See how the organism has turned this leaf a dried up brown color. And over here where the actual potato is, you can see this ring of black underneath the skin. You can also see this white powdery material on the outside of the skin. When the potatoes in Ireland got this blight, they all shrunk. They didn't grow to the right size. They were covered in this white powdery material. And if you cut them open, they were black and bad looking. They were spoiled. And again, for a long time, people thought that the culprit was a fungus. But it turns out that organism is genetically much closer to algae. than to fungus. It just goes to show you that the fungi are often confused with other organisms. They're often confused with bacteria. They're often confused with algae because they take so many different forms. It's easy to confuse them with other organisms. So, I want to address Elaine's question just for a moment, back to the honey and the Clostridium botulinum. Elaine, I cannot tell you what the risk profile is for Clostridium botulinum toxin in honey in 2021. I just don't know. I don't know that data. But I can tell you that the risk of death, if there is botulinum toxin in honey, the risk of death to an infant is high enough that that's where that recommendation comes from. In other words, it may not be common to find botulinum toxin in honey, but if it is there, it will kill the infant. So that's why we just tell parents, don't do it. Don't feed the infant under a year of age. Don't feed them honey. After a baby hits about a year of age, their gastrointestinal system grows to the point where it functions more like an adult's does, and they become less sensitive to that toxin. Now, the toxin is still, don't get me wrong, the toxin is still very potent, but the infinitesimal amount that's in honey that might be in honey wouldn't bother you and I. It might give us some gastrointestinal distress, but it wouldn't kill us. But an infant would be killed by it. And then Connie's asking about whether or not these oomycota still affect potato crops. And the answer is no, that particular organism has not been problematic for us since that time because we now have herbicides and pesticides that we can use that weren't available to the Irish farmers. So the organism is still around, but we now have modern chemicals that we can apply to our crops that would keep it at bay. I will tell you, though, that a lot of scientists are very concerned that in our country right now, most people who grow potatoes commercially only grow one species of potato. That's called monoculture, and that's dangerous from a naturalist's perspective. Another fungal organism could emerge at some point, an organism that likes to eat and destroy potatoes, and that is resistant to our chemicals. So a lot of scientists are concerned that if we ever had an emergence of a pathogen that could destroy potatoes, we'd be in a lot of trouble. We would lose all of our potatoes because they're all the same species. The reason that commercial potato farmers grow this one species of potato is because McDonald's buys it. McDonald's buys it. McDonald's is the greatest consumer of potato in our country. It's kind of gross to think of, but it's true. And so people who grow potatoes for their living want to grow the kind of potato that McDonald's wants. And McDonald's wants this one kind. So yeah, there's danger in growing only one species of anything because a microbe could emerge that creates a blight on that organism. And if we don't have the right chemical to control it, we're in trouble. Yeah, so Connie's talking about famine houses. Yeah, in Ireland. Listen, the potato famine in Ireland was, it utterly changed that nation. It utterly changed that nation. And it was all because of this little organism that grew on and destroyed their potato crop. It killed so many people. And like Connie's saying, it killed entire families. Not the organism killing the families, but starvation killing the families. That's how dependent the Irish were on this potato. And they grew it themselves in their own home gardens. And without it, they did not have that sort of basic food staple that they needed to survive. Entire families starved to death during the years of the famine. And of course, it also drove many, many tens of thousands of Irish out of Ireland because they couldn't survive. So many of them came to the United States as a result of that famine. Yeah, very historically important part of Irish history. That's right. Yeah, Marilyn's asking about monoculture with bananas. Yes, same problem. Same problem. Um, we have a problem right now with monoculture in the kind of bananas that are grown, and it's the same reason. It's because the companies, the big player companies that buy bananas around the world from banana farmers, they only want this one species of banana because they think that's what the consumer wants. And so there are some scientists who believe that this particular species of banana will go extinct because of problems related to pathogenic organisms destroying the crops. Yeah, you would think we would know by now from history that monoculture is a bad idea. If you only grow one type of a particular organism, a particular plant, if you only grow one species, you run the risk that that species is going to get wiped out at some point. It's just a bad idea. But yeah, the bananas are another great example. And the big commercial buyers of bananas like Dole's a good example. Chiquita is another one. They only want that one kind. They only want that one species. So that's what the farmers grow. All right. So a little bit of history for us there related to microbiology. The other kinds of fungus that I really want to talk about are the saprophytic fungi. Now, this word saprophytic, the other term you'll hear is saprophyte. An organism is a saprophyte if it falls into this category. And this category of fungi get their nutrients from dead things. These are the fungi that break down dead organisms. So organic materials, remember what that means to us in science. That means carbon containing. So when organisms die in the natural world, and that's whether they're plants or animals or any other kinds of organism, the saprophytic fungi are the largest group of organisms that helps break them down. And of course, we are very dependent upon them. We are very dependent on the saprophytes to get rid of all of this dead material, all of these dead organisms that would otherwise pile up on the planet. Now, saprophytic fungi, these fungi that get their nutrients from dead things, they are not generally pathogenic. They're everywhere, first of all. They're all over the environment, the saprophytes. And they're generally not a problem for us, mostly because they don't want living things. They don't like living cells. They want dead things to eat. But it is possible, and I will mention it, it is possible for people who are very severely immune compromised to develop disease from a saprophytic fungal organism. It's not common, but there are people who are severely immune compromised. Think about an AIDS patient, for example, and they are susceptible to infection with a saprophytic type of fungi. But the rest of us, they don't bother us at all. And not only do we come in contact with them on the outside of our body, but we consume them, without even knowing it every day. And again, they don't bother us. They're not pathogenic for the most part. Now, there are three big groups of saprophytic fungi, and I'd like us to be familiar with these groups. The first one is called zygomycota. The second one is ascomycota. And the third is basidiomycota. So again, this MYC term, Mike, this is the part of a word that should make you think about fungi. Mycology is the study of fungi. So zygomycota is the group of fungi that belongs to this particular family. Now, the zygomycota, a good example of one of these is the kind of mold that can grow on things like bread. But this is not the green mold that you think of when you think of bread mold. This is a black mold. So there's a type of zygomycota called Rhizopus stolonifer that loves to grow on older bread, and it grows as a black organism. This term asco actually translates as sac, and this term basidio translates as club. So the reason we broke these saprophytes down like this, zygomycota, ascomycota, basidiomycota, it's all related to what they look like. They have different forms when they grow. Now, within the ascomycota group, you'll find the yeast that's so important to us in terms of some kinds of food production. The Saccharomyces cerevisiae, that's a mouthful. Saccharomyces cerevisiae is also called brewer's yeast. These are also the dry kinds of yeast that you buy to make bread. So these yeast are very helpful to us. These ascomycota yeast are very helpful to us in the production of some foods. And finally, the basidiomycota, these are the club fungi. Penicillium is in this group. Now, penicillium is that green mold that sometimes grows on your breads and other foods. And penicillium, of course, is sort of famous because it's from penicillium that we get penicillin and other antibiotics related to penicillin. So this is a type of fungus that produces an antibiotic to protect itself from other fungi that might want to harm it and bacteria that might want to harm it. Another type of basidiomycota of interest is a type called Aspergillus. You may have heard of Aspergillus. Aspergillus is a fungus that's a real problem for farmers. Aspergillus likes to grow on grain. So in big storehouses of grain, think about silos that are full of wheat or full of corn. And these are the grains that are, they're dried and they're stored until they can be sold. Aspergillus loves to grow on the surface of these dried grains. And like that Clostridium we were talking about earlier, Aspergillus makes a toxin. So sometimes, if farmers are not careful, they can get a big growth of aspergillus on, again, stockpiles of grain. And that aspergillus will secrete toxin that gets onto the grain and can be toxic. It doesn't tend to kill people outright. You have to eat a lot of it for it to kill you, but it can cause other sort of chronic health problems for people. So farmers have to be very careful not to allow this fungus to grow in their barns and their storehouses. All right, so just a little bit of mycology for us today. The saprophytic fungi that we depend on so much to degrade dead organisms. And there are 3 particular types that are just practically important to us. Okay, any questions? Any questions about anything we've been talking about? You know, there are other organisms that help degrade dead things. There are bacteria, for example, that help break down dead materials, dead bodies, essentially, dead plants, dead organic matter. But the saprophytes are critically important to that process. If it weren't for the saprophytes, if the saprophytes were suddenly removed from planet Earth, we'd be in trouble. We would be in trouble because it would take a lot longer for those things to degrade in the natural environment. All right. So I want to switch gears now and talk a little bit about the microscopic types of fungi. We're going to go into the microbiology aspect of it now. Remember, we think of a microbe as being microscopic and also single-celled. When it comes to microscopic fungi, there are 2 kinds. There are yeasts and there are molds. Now, when I use those terms, I'm not talking about a particular species of fungus. I'm talking about a particular form of fungus. Remember, the fungi are very diverse and they tend to have very complicated life cycles. And for many of them, there's a time in their life cycle when they exist as a microscopic form. That means they either exist as yeast or as mold. Those are the 2 forms of fungi that are microscopic. Now, let's start with the yeasts. Yeasts are true microbes because not only are they microscopic, but they're also single-celled. So that form of a fungus is a true microbe. We've seen yeast before in our lab. We've seen what they look like under the microscope. Remember, they're sort of spherical, maybe a little more egg-shaped or oval-shaped. Yeasts are found all over the natural environment. Remember, we saw them when I took a strawberry from the grocery store and I just mashed it up in a dish with a little bit of water. That strawberry was covered in yeast. And when I put a little water in there and I opened up the strawberry, I released the sugars in the strawberry, those yeasts were able to grow. So yeasts are very, very common. They're all over the environment. Many, many different species of fungi can take the form of yeast. Now, yeasts are made, if you remember from that lab, yeasts are produced through an asexual process. One yeast cell is going to create offspring through a process called budding. And what happens is a very small, brand new daughter cell is going to bud or emerge off of the surface of the parent cell. And that little bud is going to grow and grow until it's a full-grown adult yeast cell. And then that daughter cell will break off of the parent cell eventually. But it's important to note that sometimes when you watch yeast out in the environment, when you watch them reproducing like that through budding, sometimes the new daughter cells don't break off right away, and you end up with this long branch of yeast cells. One cell produces the next, which produces the next, which produces the next, and you get this long chain of yeast cells. Under the microscope, a chain like this can look like a mold. Let me say that again because that's important. Under the microscope, when we get long chains of yeast cells produced through budding, they can look like those branches that we saw with mold. Remember, those are called hyphae. So when you have a chain of yeast cells that fails to break apart into individual cells, we call those pseudo hyphae. They're not true hyphae. They're not real branches of mold, but they look like them under the microscope. I'll show you what I mean in just a minute. On this slide in the upper left-hand corner, you're looking at some yeast underneath a scanning electron microscope. Remember, the SEM, the scanning electron microscope, is going to show us the surface of cells under high power magnification. So you're looking at several yeast cells here. Look at this one right here and this one right here. These are two yeast cells, two what we could call parent cells that are reproducing. They are using an asexual reproduction process called budding to create a new cell. The new cell is going to be genetically identical to the parent. It's just copying itself, it's cloning itself. And this little bud is going to keep getting bigger and bigger, and then it's going to break off and be its own independent cell. Same with this one down here. It's budding off of the parent cell. It's going to grow and grow. It's going to break off and become its own unique individual cell. Sometimes, though, the budding process creates these chains of offspring. See that right there? This parent cell right here will start to bud. Here's the emerging bud. And then instead of this one breaking off, the process repeats and repeats and repeats, and you get this chain. This is what we would call a pseudo hyphae. It looks like a hyphae. It looks like one of the branches that we see in mold, but it's not. It's actually a chain of yeast cells. This image over here on the right, this is one that you saw from that mashed up strawberry. We're under the 100x lens here. So we're at 1000x total magnification. And this is the fungus that was growing on the strawberry. You can see hyphae here. Those are the mold branches. And you can also see chains of yeast cells, pseudohyphae. So this particular fungus is taking both forms under the microscope. It's both a mold and a yeast. And like I said, that's not unusual. Lots of fungus can grow as both mold and yeast. Some of them only grow as mold. Some of them only grow as yeast. Some of them grow as both. Okay. Now remember, I said one of the things that makes the yeast unique is that some of them are aerobic, but many of them are anaerobic. They do their metabolic processes without oxygen. Different species of yeast, in fact, can be identified in the microbiology lab by testing their metabolic abilities. For example, a lot of yeast are classified as facultative anaerobes. Now, what does that mean? It means that they're anaerobic. They don't use oxygen primarily, but they're also facultative. They can switch and swap back and forth between an oxygen-rich environment and an environment that doesn't have any oxygen. And it doesn't bother them because they can switch metabolic pathways depending on where they are. So a facultative anaerobe is going to use oxygen if it's available to do its primary metabolic pathways, including making ATP for energy. In other words, a facultative anaerobe type of yeast, if it's in an oxygen-rich environment, it's going to do aerobic cellular respiration. But if it finds itself in a place where there's no oxygen available, well, that's okay. It's not going to die. It can just switch over to an anaerobic form of cellular respiration, or it can switch to a fermentation pathway. We actually like fermentative yeasts, don't we? We use fermentative yeasts to make a lot of the food that we like to eat. I mentioned the kind of yeast we use to bake our breads. We also use yeasts in producing certain beverages, especially alcoholic beverages. We also use yeasts in fermentation to make certain industrial chemicals. We love yeast because yeast makes products that we like to use. So, genetic diversity. The fungi are very diverse. They're diverse in how they look, what their morphology is, and they're also very diverse metabolically speaking. When it comes to the fungi, they can do combinations of metabolism that you and I can't do. They can do aerobic cellular respiration. They can do anaerobic cellular respiration. They can do fermentation. Some of them can do all three. It's amazing. They're much more adaptive than you and I are. You and I are much more limited in what kind of metabolism we're capable of. Now, I want to spend just a couple minutes talking about the molds. And when it comes to the molds, it's primarily vocabulary. Both yeast and mold are the two forms of microscopic fungi. So remember, we're not talking about individual species of fungi here. We're talking about life forms. Many species of fungi can grow, at least during part of their life cycle, as a mold. Now, a mold is microscopic, but it's not single-celled. A mold is multicellular. That's an important distinction. The yeasts are microscopic and single-celled. The molds are microscopic and multi-celled. That means that molds are not technically microbes. These are multicellular organisms. And those many cells grow as branching filamentous strands that we call hyphae. So when you see a mold under the microscope, it looks like a little bush or a tree with lots of branches. And those filamentous strands are called hyphae. Now, there are two kinds of hyphae I want us to be familiar with because you can recognize them under the microscope. The first one is called septate hyphae, and the second one is called coenocytic. This is a terrible word to spell. Coenocytic hyphae. What's the difference? Well, in septate hyphae, each individual cell in the hyphae is separated by a wall, or what we call a septum. So when you look at a branch or a hyphae, you can see each individual cell separate from the others. In a coenocytic hyphae, you don't see the individual cells because there's no wall in between them. In other words, the hyphae look like one long continuous structure, even though there are multiple cells inside. Now, when you grow fungi in the laboratory, and we do all the time in the microbiology laboratory, you can grow vegetative hyphae or cellular hyphae both on growth media and in growth media. So in other words, you can grow molds on the surface of agar or in a liquid broth. It just depends on the individual species, which one is a better way to grow it. Now, when we grow mold on the surface of an agar plate, for example, it's first going to grow right on the surface, just like bacteria do. But if you leave it, if you allow it to grow for longer periods of time, say several days, what's going to happen is it's going to start to grow upwards. It's going to start to grow up above the surface. At that point, it's going to look very fuzzy. It's going to look like what we think of mold as. And at that point, it's preparing to reproduce. So when you first grow a mold on agar, it's going to grow very flat, the same way bacteria does. After a few days, it's going to start to grow upwards off the surface of the agar, above the agar. Those hyphae that are growing up, those fuzzy looking hyphae that are growing up off the surface, those are reproductive. And those are the structures that are going to make spores, reproductive spores, to shed out into the environment. On this slide, you're looking at some drawings on the left-hand side and a couple of light microscope images on the right. What the drawing is showing us is the difference between septate hyphae and coenocytic hyphae. So this structure right here that looks like the branch that you might pull off a tree, this is a mold. It's a multicellular organism. And those cells join together into these branches or hyphae. This particular mold produces a septate hyphae because we can see each individual cell. We can see the boundaries between the cells. The wall that separates one cell from another is called a septum. Now compare that to the mold on the bottom of the drawing, which is a coenocytic hyphae. Notice that there's no septum separating each cell. I can't tell you where one cell ends and the next cell begins. I can still see that it's a multicellular organism though, because look, there are multiple nuclei. Every once in a while, I can see a nucleus. There's one nucleus per cell. So I know that there are many cells in this mold. I just can't see where they begin and end. That's a coenocytic hyphae. And this is what that looks like under the microscope. This is septate hyphae that have been stained with crystal violet. See the septum in between? You can tell where one cell ends and the next cell begins. These are septate hyphae. Now, compare that mold with this one. This is a coenocytic mold. Take a look at the hyphae. I don't see any septum. I don't see individual cells. But look, here's a nucleus right here, that white area. Here's another nucleus right here. There may be another nucleus right here. So I've got multiple cells here. I just can't tell where one cell ends and another cell begins. I just can't tell because there's no septum. So this is the septate hyphae, and this is the coenocytic hyphae. Two types of mold. One of the things that I find very interesting about mold is that mold is a microscopic organism or a form of fungus that, if left to its own, can sometimes become macroscopic. Molds can grow and grow and grow to the point where we can see them with our naked eye. Probably the easiest place to see it is in your bathroom, right? We all have seen mold growing in our bathroom before. We like to call this mildew. That's another name that gets attached to this particular organism. You've probably seen it growing in between tiles, on grout lines. It likes to grow around the edges of bathtubs. It's mold. And when it first started, it started microscopically. We wouldn't be able to see it. But as it grows and grows and grows and grows and grows, it becomes macroscopic. So that's a pretty interesting organism. That's a form that fungi can take that can grow so big that we can see it with our naked eye. All right. Oh, Elaine's asking about, are there some cells that have multiple nuclei? Is it possible to have one cell that has many nuclei in it? And the answer is yes. They're not very common. They're, of course, eukaryotic because only eukaryotes have nuclei. But yes, there are certain cells in the human body that one cell can have more than one nucleus in it. Now, the way those cells come about is because young forms of the cell, generally stem cells, will merge together and create one big cell. They're fascinating. You can see some of these in bone. You may have learned about certain types of cells in bone that are multinucleated. Early on in the development of that cell, so back when it was just a stem cell, several stem cells joined together and they all brought their own nucleus, and they came together and merged and formed that one multinucleated cell. Yeah, great question. And yes, that is a feature of eukaryotes. Now I'm just looking at Marilyn's question. Yeah. So Marilyn's talking about a famous New York City bakery that made jam and jelly and batched it in barrels. So made huge barrels full of these jellies And of course, molds grew in it, because of course they did, right? Fungus grew in it. And apparently, Marilyn's saying they used to just, cut off the moldy bits and sell the rest of it. I love those stories. If it's not a pathogenic mold, it's not going to hurt you. I imagine it probably gives the jelly a unique taste, but to each his own. If you like that taste, probably not a problem. That's a great story. And certainly, there are lots and lots of fungi that we eat all the time, right? I mean, I know I love mushrooms. And there are all kinds of food products that are made from molds and made from yeasts. So yeah, most of them are not harmful. Remember, the vast, vast majority of microbial cells are not harmful to us. They're not going to cause us any problems at all. So yeah, here's a picture just to remind us all that molds can become macroscopic. Here's somebody's bathtub with tiling around it, and you can see where the mold likes to grow. Molds like to grow in cracks and crevices that tend to stay wet. They love bathrooms because bathrooms tend to stay warm and wet. So how do we know what kind of mold we're growing in a laboratory? Like, how do we know if a sample of mold comes in? How do we know what genus and species it is? Well, we can go by several different features. We can grow mold on an agar plate, like I mentioned, or even in a broth tube. If you grow it on an agar plate, you can look at its colony morphology. Just like we talk about bacterial colony morphology, we can also talk about fungal colony morphology. Under the microscope, we can look at its hyphae. What does the hyphal morphology look like? We can look at those reproductive spores, because sometimes their body form, their morphology will give us clues as to what species we're looking at. The last thing that has become very common in identifying individual species of mold is by looking at the sequence of bases in their ribosomal RNA. Remember, rRNA, that's ribosomal RNA. These are the pieces of RNA that are found inside the ribosome. And just like all cells, fungal cells have ribosomes. So by looking at that sequence and comparing it to a database of our RNA sequences, we can identify different species of mold. So here's that term mycelium again. Remember, when a mold grows to the point where you can see it with your naked eye, now it's called a mycelium. And mycelia out in nature tend to grow in the soil. Any fungi that can grow as both a yeast or a mold at different times in its life cycle, those are referred to as dimorphic fungi. Dimorphic translates as two forms. So these are fungi that in their certain points in their life cycle can grow as either a yeast or a mold. Now, it is possible for fungi to cause clinical disease in humans and other animals, but it's not very common. We just don't see fungal infections as often as we see bacterial and viral. Remember, when it comes to pathogens, microbial pathogens, the greatest culprits are bacteria and viruses. But there are some species of fungi that can cause clinical disease in people. Typically, it's due to some other problem going on. And one of the problems that can give someone a fungal infection is a problem with their microbiome. You may be familiar with the kinds of yeast infections that humans can get. They're not very serious infections, generally speaking. People can grow yeast really anywhere on their skin. They can grow yeast in their mouths. That happens sometimes. They can grow yeast in their urogenital regions. Women get vaginal yeast infections from time to time. Again, these are organisms that are part of the microbiome. So normally they don't cause us a problem. But if there's a disruption in our microbiome, sometimes the yeasts will overgrow. They take advantage of this disruption in our normal microbiome and they sort of bloom. So when, for example, a person gets a yeast infection on their feet, what we call athlete's foot, that's coming from yeast that belong on the skin. They belong there. They normally live there, but they will overgrow when there's a disruption in the skin microbiome. And that disruption usually comes from wearing dirty socks, not changing your socks often enough, leaving wet and sweaty socks on for too long. Yuck, right? Athlete's foot. When it comes to yeast infections of the urogenital tract, think about vaginal yeast infections. Those are often secondary to a disruption in the microbiome inside the vagina. There are normally yeast in the vagina, but if a woman has some kind of disruption to her microbiome, maybe she's taking antibiotics, maybe she's had some kind of a hygiene related issue, that can cause the yeast to overgrow and you'll end up with an infection. The good news is that if you can get the microbiome restored, that yeast infection will generally resolve. And if not, you can also give that patient an antifungal medication, usually a cream or something. We generally use topical cream forms of antifungal for those kinds of infections, but they don't tend to be serious. The one time that we do see serious fungal infections is in people who are immune compromised. It is possible, for example, for a person to get a fungal pneumonia if they are sufficiently compromised. Their immune system is either busy battling another infection or their immune system isn't working normally. So again, in an AIDS patient, in a cancer patient who is undergoing strenuous chemotherapy, in patients who are otherwise severely ill, think about a burned patient, then we can sometimes see more serious fungal diseases, fungal infections. This image here on this screen, this shows a chest x-ray from a person with a fungal pneumonia. So just to orient us, this is the mediastinum right here, right down the center. Here's the shadow of the heart. This structure right here is the diaphragm, so the abdomen would be below it. You can just see the shadows of the ribs across the lungs, and the lungs are the black spaces, the air-filled spaces. Here's the collarbone, here's the other collarbone. The person's head would be up here. Now, what I want you to notice is that these black spaces are not purely black. There are these little white dots in there. Can you see them? That's fungi. Those are fungal organisms growing inside this person's lungs. Again, not common. If we see a fungal pneumonia in a patient, a human or another animal, we want to know what else is happening that has compromised that person's immune system to the point where they weren't able to fight that off. Yeah. Now, in the laboratory, when we grow fungi, we have to be kind of careful because remember, if we grow the fungi on an agar plate, if we let them grow for too long, they're going to start to grow above the agar plate and they're going to start producing reproductive spores. And they're going to release those spores into the air. So just like when we were children walking through the forest and stepping on different mushrooms to release spores, we have to be careful that we don't release spores into the air in a microbiology lab. They can be real problematic for us. We grow fungi on agar that is specifically designed for fungi. So we take those agar crystals that we talked about and we add water, and we also add in things that will help fungus grow and prevent bacteria from growing. If we're going to grow a fungus, we want to make sure that we take extra steps to prevent any bacterial contaminants from growing should they land on that plate. So generally speaking, when we grow a fungus, we use a special kind of agar, and I have the name of it right here. It's called Sabarodes agar. It's a bad word to spell. Sabarodes. Sabarodes has a very short list of ingredients in it. It's basically glucose and peptone in water and agar. Remember, the glucose, that's the carbohydrate, that's the energy source. And peptone, that's that partially digested protein, that's the source of carbon for building other molecules. So it's a very simple, very basic, what we would call chemically defined culture media, and it's specifically for fungi. We also keep the pH of Sabarodes agar low because that's going to help inhibit bacterial growth. We have lots and lots of growth media in the microbiology lab. Some of them are designed to grow bacteria. Some of them, like Sabarodes, are designed to grow fungi. And by playing around with the ingredients in the media, we can meet our own needs. I want to grow fungi, but I don't want to grow bacteria accidentally. Well, I can adjust the pH of that agar so that just in case any bacteria land on that plate, they're not going to be able to grow. All right. Any questions? Any questions about yeast versus mold? Microscopic versus macroscopic mold? Septate hyphae, coenocytic hyphae? How we grow fungi in the lab. All right. So the last few slides today are an exercise for us. I grew some fungi for us at home. I didn't use my strawberry method this time. I used a slightly different method so that we could see fungus growing on a surface. I didn't grow it on Sabarodes agar. I used a more natural surface, and that's what we'll look at next. So what you're looking at in this image is a glass dish that has pears in it. These are the kinds of pears that you can buy in a can at the grocery store. You probably recognize them. These are Bartlett pears. These pears come halved and skinned. And you can either buy them in what's called a heavy syrup or a light syrup. The heavy syrup has extra sugar added to it. And the light syrup is generally just a fruit juice. These happen to have been light syrup pears, happen to be what I had in the pantry at the time. But all I did was I took the pears out of the can, I laid them into this baking dish with a little bit of that light syrup. And I left them on the countertop. And this is what happened within a few days. I believe this was about 3 days. So what you're looking at here are fungal organisms growing as colonies on the surface of this fruit. Remember, a colony is just one cell that landed on that surface and then divided lots and lots and lots of times until it became a visible clump of cells. And those cells are all clones of each other. They're all genetically identical. So putting that fruit in the open air allowed fungal spores to land on the fruit, return to a vegetative state, and start to grow, start to bud. When you look at a colony on an agar plate, you cannot tell whether it's a bacterium or a fungus. Hopefully you've had a chance to look at that lab supplement video about colony morphology. And one of the things we talk about in that video is how fungal colonies and bacterial colonies can look alike. So you can't tell just by looking at it whether it's a fungus or a bacterium. You have to take a sample and look under the microscope. These are just colonies. They're just groups growing on the pears. Now, that dish was open to the air. I don't see bacteria growing on here. Why? How come those pears aren't just covered in some kind of bacterial growth? Why is it fungus that grew instead? I've got both microbes in the air. I've got bacteria in the air. I've got archaea in the air. I've got fungal spores in the air. So how come only the fungi grew? Yep, you've got it. Connie's got it. Valerie's got it. Those pears are sweet. Pears are sweet anyway because they're fruit, but canned pears are sort of artificially sweet. They've been skinned, the skin has been removed, so only that sweet flesh is exposed, and it's been soaked in sugar water. It's been soaked in fruit juice. and sugar syrup, right? It's very high in carbohydrate, simple sugar. That's going to bind up available water. It's not the amount of water. There's plenty of water in a can of pears, right? But most of that water is unavailable. Bacteria have a high requirement for available water. They just can't grow well on those pears. The fungi don't have that high requirement. They'll happily grow on the surface of those pears. Now, Connie, your point about acidity is a good one. These are pears, so it turns out they're not particularly acidic themselves. You're right, sometimes ascorbic acid or vitamin C is added to canned fruits, but not to these pears. So this is a case more about carbohydrate and water availability. Now I left these pears and I let these colonies do whatever they were going to do. Now notice that some of the colonies are really pure white, but I've got some colonies that are growing as green. That's simply because I have different species here. I have multiple species of fungi growing here. And I can tell that because of the morphology of the colony. This colony looks very different than from, say, this colony. Take a look at the dish as it looked after about 6 days. Wow, this looks very different from this. These are colonies growing on the surface of the pear. After a few days, they're going to start growing up. The hyphae are going to start to grow up off the surface and become reproductive. That's when you start getting the fuzzy look. I took a plastic tote box, basically, and I covered this dish. There was enough air in there and enough circulation in there to allow those fungi to continue to grow. But I didn't want a whole lot of the reproductive spores flying around in my kitchen, so I did keep a big dome over that dish. But see what's happening. That fungus is doing great. It's growing great. It has reproduced and reproduced, and now it's ready to send spores out into the world to spread itself to other locations. That's why the hyphae are growing up. And the spores are going to be attached to those hyphae. And you get that cottony kind of look, that real fluffy kind of look to the fungus. Notice that where you can see the colonies, they've really gotten big. Some of them are black. Some of them are green. Take a look. Look at that big, huge colony. Again, I've got different species of fungi growing here. I did want to take a minute and talk about a colony perhaps that looks like this. I can see this with my eyes. This is a mold or a mycelium. Remember, the molds are microscopic, but they can grow into a macroscopic mycelium. There are some people who see a black color to mold and panic because they think it is the notorious black mold. There is a type of mold called Staphybotrys. That's the genus name. You don't need to write it down. Staphybotrys is a very dangerous, toxic type of fungus that grows with a black color to the mold. There are many, many other species of fungus that grow black, though. So just because a mold is black does not mean that it is Staphybotrys. Lots of mold grows with a black color. Most of the black mold that you can see in nature is not that horrible toxic Staphybotrys. So lots and lots of fluff growing on these pears. I did want to just mention a little trick that we use in the microbiology laboratory. I wanted to transfer some of this material onto some Sabarodes agar, and that's what you're looking at here. This is a plastic petri dish with Sabarodes agar in it. And what I did was I took a couple of colonies. I chose the green organism, and I placed those on the dish. And then I also wanted to take some fluff, so I placed that on the dish. And of course, I had to be silly because I'm that way. So I made a smiley face on the dish. You can transfer mold from the environment into the laboratory into culture dishes. I could also have taken some of the material and put it into a broth culture if I wanted. But since it was already growing on a solid surface, I chose to put it onto a solid surface in the lab. Now I want you to look at my fingers in the picture here on the left. If you want to take a sample of the kind of hyphae that grow upwards, the fluffy kinds of hyphae, it's really hard to grasp onto them because they're so delicate and fragile. A little trick that I learned when I was in veterinary practice was to take a piece of Scotch tape and make a loop out of it with the sticky side out and just touch the tape to those fluffy clouds of hyphae. Then you take the tape and you press it onto a glass slide. And you'll be able to see the hyphae under the microscope. It's just a little trick. It's an easy way to gather some hyphae. You get them to stick onto this Scotch tape, and then you just press the tape down onto a slide and then remove it. And it will leave those hyphae on the slide. Remember, we can use the structure of hyphae to help us identify a fungus if we look at them under the microscope. All right. I only have one more slide to show us, so I'm going to skip right to it so I can get you guys out in a reasonable amount of time. This is a clinical question, a clinical application. This sample that you're looking at here came from a person who had a fungal infection. This happened to be a yeast infection of the vagina, so a vaginal infection with fungus. When we took the sample and we spread it out onto Sabarodes agar, we got these colonies growing. Again, if I saw just this, I would not know that this was a fungus or a bacterium. I would have to do a little bit more examination. I'd have to make a smear and look under the microscope. Okay, you can't tell just by looking what you've got. Over here, this is what we saw on the smear. Now, this smear, that as you're looking at it right here, is under the 20x objective lens. How do I know that these aren't bacteria? Could those be bacteria? They They look like bacteria. They picked up stain. They look like maybe little fat little rods, maybe, or coxobacilli. Yeah, Elaine's got it. Connie's got it. I'm not going to see bacteria on the 20x lens. Not going to see them. And look how big they are. They're huge on the 20x lens. Yeah, these cells are too big to be bacteria. Eukaryotic cells are big. These are yeast. And this happens to be a very common genus of yeast called Candida. Candida is a super common yeast that is found on human beings, on and in human beings and other animals. But this happens to be a pathogenic Candida. This is one called Candida auris, O-U-R-I-S. Candida, again, the genus Candida, has many different species within it. And most Candida does not harm us. Again, you can get a Candida infection if your microbiome is a little out of whack. You can get a Candida infection, but it's easy to treat. This Candida auris, this one is a pathogen. And what makes it kind of scary is that we're seeing more and more of it. There are more reports from all over the world of patients who are coming down with infections with Candida auris. And they're a little hard to treat because it's a pathogen. So it's a reminder to us that we always have to be on the lookout for emerging pathogens. Sometimes we'll get so used to an organism being non-pathogenic that we forget that organisms change over time with mutations. And this particular species of Candida is a newer pathogen and one that is tending to cause more and more disease over time. All right, very good. All right, any questions about anything we've talked about today? Just a little overview of the fungi. I hope maybe I've sparked a little interest in them. They really are fascinating. Just like the bacteria, most of them are harmless. Many of them are helpful. All right. I'm going to let you go. I will see you on Wednesday. We're going to talk about a certain category of culture media called the special media. And I will see you then. All right, everybody. Enjoy the rest of the day.